This Week in Virology, the podcast about viruses, the kind that make you sick. Welcome to This Week in Virology, episode number 92, recorded on July 20th, 2010. We're live from the 29th annual meeting of the American Society for Virology in beautiful Bozeman, Montana, on the campus of Montana State University in the wonderfully named Procrastinator Theater. Okay, I'm Vincent Racaniello, and as always, we have a great crew to give you your weekly dose of viruses, starting with my TWiV colleague on the left here from the Department of Microbiology and Molecular Genetics, the University of Florida, Rich Condit.
Hello, Vincent. Here we are again face-to-face. This is pretty weird.
Yeah, usually we don't do this face-to-face. Rich doesn't fly up every Friday. We do it via Skype, the voice-over internet protocol. Also joining us today are special guests over on the left, Karla Kirkegaard, Professor and Chair of the Department of Microbiology and Immunology, Stanford University School of Medicine.
Hi, Vinny.
Welcome.
Thank you.
And next to Karla, Marilyn Roosinck, Professor at the Samuel Roberts Noble Foundation. Welcome to TWiV.
Thanks. Hi, Vince and Rich.
And of course, we have today an amazing audience of virus addicts. More than we've ever had. We've done a few live TWiVs. The first one, we had one audience member. The second, at the University of Florida, we had about 20. And now I would say, since this room holds 200, we have 200 people, right? Yeah. Of all places, if we couldn't get an audience here, where could we? So thanks everybody for coming. We're going to try and keep it tight today. Usually we don't have time constraints on TWiV, so this is a little different for us. But we're going to give you a flavor of what this is. And what is this? So every Friday we record a podcast. A podcast is simply audio content that you record and you put on the internet. So anyone can make one and anyone can download it. So it makes dissemination of information incredibly powerful. We started this in the fall of 2008. A colleague of mine at Columbia, Dixon Despommier, who was supposed to be here and couldn't make it, we began. A few months later, Alan Dove, a science writer, joined us. And at the end of 2009, Rich Condit joined us. So we have a permanent crew of what we call hosts for the show, and then we have guests on some shows, or on other shows we talk about the week's stories in virology. The goal of this is to make virology accessible to everyone. And we really didn't know who the audience would be when we started it. Now, obviously, many of you are fans. Who listens to TWiV? Can we see? So about half, right?
Like that, yeah.
Something like half. And here you're learning about it. But the amazing thing, and you'll see from the emails that we read, is that many, many other people love it because they can learn about what we do. And I think that's very powerful. That's why the podcast format is so powerful. They can understand not only the science, but how science is done and how virologists interact. By the way, you can interrupt me at any time, guys, because I'll keep talking. All right?
Good.
And just to give you an idea, each episode, so we've now done 92 episodes. We're reaching the 500,000 download mark since episode 20, I think, right?
Yeah.
That's about 7,000 downloads per episode. That's amazing. That's more people I've taught in my whole career per week. So that's the amazing power of the podcast.
And we get email from all over the world.
We get email from all over the world, from all ages and all works of life, not just professionals, but all kinds of people. I got an email a while ago from a guy who paints houses and he listens to TWiV while he paints the house. Okay? So how much cooler can that be to disseminate this kind of information? So this is very powerful and very compelling. And of course, you're all part of it because someday you will be on TWiV as well. So what we thought we would do today is chat with Karla and Marilyn about the work they've talked about at this meeting. And remember, we'll probably be a little repetitive because people will be listening to this other than you. Then we'll do a few emails to give you a flavor of the kind of interaction. That's really the best part for me of this whole thing, besides learning more than I would ever learn in my own career, hearing the people interacting, the questions that they have, and how much they're interested in science. I think that's the best part. Would you agree?
I would agree, absolutely.
So you'll hear some of those emails later. And then at the end, what we typically do on TWiV is we do a science pick of the week. Each of us picks a book, a website, an article, a museum, anything science-related that we'd like to share with people. We keep an archive of all of this. There's a website, TWiV.tv, where you can find it. You can find all the episodes, all the picks of the week, and all the letters are archived as well. So that's to give you a little idea. There is a session in here at 1:30, so we have to be done by 1:15, and we'll certainly try and do that.
Vince, what are people's interests for the most part? I mean, I remember about 10 years ago when I was teaching undergraduates, and every single one came in and said, I read The Hot Zone, and I really want to work on viruses.
Yeah, a lot of people do. And in fact, people tell me that they have begun a career in virology because of listening to this.
One of those is sitting right there, Eric Donaldson, who's been on the show before.
Right.
Got his start reading The Hot Zone.
Is that right? Yeah, so I'm sure many of you have similar experiences. Many of you, in fact, have been on TWiV. But people are not, I think that's what fascinates people with viruses to begin with, the kind that make you sick.
Yeah.
So, the tagline for TWiV is the netcast about viruses, the kind that make you sick. Now, after listening to Marilyn, I realized it's probably not a good idea, right? But I wanted to distinguish it from the zillions of podcasts out there about computer viruses. And so I thought that would be neat to remind people this is not about computer viruses, but about the biological kind. But in fact, we get lots of IT people listening. And they are really interested in modeling virus mutation and evolution.
So maybe you should change it to the kind that don't make your viruses sick.
The kind that don't make your viruses sick. And in fact, that's a great lead-in to what I wanted to ask you about. So you told us the other day about how viruses actually can make plants healthier.
Mm-hmm. Yes.
And we had actually covered the grass Yellowstone story on an old TWiV
right.
a long time ago, and actually a listener alerted us to that. So one of our listeners reads your papers, in fact. So how pervasive is that?
Well, I think probably most viruses don't make us sick. I guess my estimate is about 1% of viruses are pathogenic. And that's based on really old work that most of you are too young to remember. But, SV40, most people have heard of SV40, right? But have you heard of SV1 through 72? No, because they didn't make anything sick. But when they were when they were starting to do cell culture, they got all these simian viruses coming out of the cell cultures. And SV40 was the only one that made anybody sick, and so that's the only one anyone ever studied. So that's around 1%. That's my estimate.
So that's all viruses, not just plant viruses.
Right. All viruses. So for plant viruses, when we find viruses in the wild in plants, they don't make the plants sick. We haven't found any really symptomatic viruses in wild plants. I'm not saying there aren't any, but in our surveys so far, we haven't found any. But they're all full of viruses,
So when we eat vegetables and fruits, are we consuming a lot of viruses?
Lots. Yeah, great extra protein, little extra RNA. And if you go to a health food store, you can buy RNA capsules. I just say eat more plant viruses.
So are these of any potential harm to us at all?
No. If they were, we've been doing this. Humans have been eating plant viruses since humans were first on the earth, so I don't think so.
We covered a paper on TWiV a while ago in which we talked about a paper originating from France where we went to the supermarket and they bought 20 different products containing chilies. Probably many of you, maybe the person who did it is actually here, I don't know. But then they found that most of them contained chili pepper mild model virus was the virus.
Yeah.
And they actually tried to correlate gastrointestinal symptoms with the quantity of virus being excreted.
And was there any correlation?
There was an association, but who knows what that means.
It's probably
right?
because those people were eating a lot more chili peppers.
That's kind of the
not
conclusion.
because of the virus.
Yeah, I think so.
It was pretty weak.
Well, I think the more spicy stuff you eat, the sicker you feel, and that's the correlation.
Yeah.
But they actually found some antibodies in the serum of some. So is that likely to happen? You think?
Probably, yeah. I mean, the load of viruses that you eat every day is probably quite high. So it's not surprising that you might have a few antibodies.
One of the striking things I also got from your talk was that we have all these agricultural crops that are sickly in many cases because they don't have the right viruses.
Well, or they don't have the right microbes. Wild plants that look really healthy are always full of microbes, not just viruses, but bacteria and fungi too. And mostly in agriculture, they try to keep everything really sterile. They dump a lot of chemicals on the soil to kill all the natural microbes in it. And yeah, so it's something that we need to think about in terms of agriculture.
So Marilyn, that was my question: is how do you keep a plant sterile? I mean, you could put antibiotics around, but
right. They're not really sterile. I mean, because they carry endophytes that are carried through the seed, and so they're not really sterile. But there's an effort to keep them from having bacterial and fungal and viral
So is it
infections.
that, or is it the monoculture, that they're not getting cross-infected, if you will, from other plants that are living in the same?
So monoculture is another element of that. I think it's a combination, because when you have a monoculture, and if you happen to get a virus that is really virulent in a plant, and you have thousands of acres of monoculture, it's going to wipe it out, because it's going to have so many hosts available. So it's a mixture. I think it's a mixture.
So a question occurred to me listening to your talk. We have a sort of a dogma in animal virology that comes under the umbrella of zoonoses, which is a sort of an anthropocentric concept of when viruses from other animals infect us as if we weren't animals, okay? They might cause significant problems because we're unfamiliar with them, and we're in disequilibrium with these viruses. But then over time, if they can establish transmission in humans or another species than their own, they establish an equilibrium. And I'm wondering if the same thing happens in plants, where there's cross-species transmission, and whether that has any consequences for the pathology.
So with plant viruses, it varies a lot. Some of the viruses, cucumber mosaic virus infects 1,200 different species, so it has a huge host range. And then other plant viruses are specialists. But moving from wild plants to crop plants is something that we're just starting to study. So we've done some work in Costa Rica on wild plants, and we find mostly brand new viruses that that are not in any databases, and they're kind of related to known ones. But we have found a widespread infection of a virus that's a pathogen in the crops right nearby, and in the wild plants there's no symptoms. So I'm not sure why yet. We're trying to look at that more carefully, but there certainly is movement between crops and wildlands, and maybe it might be a benefit actually to be closer to wildlands, because not only do you get, the viruses in the wild plants are not symptomatic, but also then you have a lot of beneficial insects and things that keep the virus transmission lower too. Do
you think that at some point in the future we would be actually introducing viruses to crops to make them healthier?
I hope so. I think it'd be great.
The first virus studied was tobacco mosaic virus, right? Now, how did that show up? What's the history?
That was disease. So the tobacco had a mosaic disease, green and yellow.
But it's a cultivated plant, right?
It is a cultivated plant, yeah. And it was filterable. That's why they knew it wasn't a bacteria. They thought it was a toxin at first, and eventually they realized it was an infectious agent.
Does TMV ordinarily hang out in tobacco, or does it have another host?
It has a few other hosts. In tobacco, a lot of the tobacco cultivars that are grown are resistant to TMV, but it's still there in a lot of them. And if anyone in here smokes, I can promise you have TMV on your hands and your clothes, and if you grow tomatoes, you might make them really sick from that. It's one of the most stable viruses known. So
if you're a smoker, you're going to really worry about making your tomatoes sick, right?
You should, yeah.
So what do we need to know before we could? So 2 questions. What do we need to know before we could put viruses or maybe other microbes into crops? And maybe more importantly, would people eat crops that have viruses in them? Because, GM food is makes a big controversy. Is that going to be an issue?
I think it's going to be less of an issue. First of all, they're eating food with viruses in it all the time anyway. Not just plants, but animals too.
But they don't know that.
No, well, so maybe we don't have to tell them. No, I think it should be less of an issue than GMOs because it's not doing anything that's not already happening. Although you could argue GMOs are sort of already happening too, but
Yeah.
I'm really excited about all this microbiome stuff that goes on because I think a lot of people figure that they're sterile, all right? And I think it's important for people to understand through your work and through other work that are looking at microbiomes that we're crawling with bugs all the time. You know,
Yeah.
it's important.
yeah. And I think Julie Pfeiffer in her talk yesterday mentioned that we're about 10% human as far as cell counts go. I've heard actually as low as 1% by some people, but
That was dinner conversation last night. We didn't know whether to be insulted by that or not.
I just say good thing our cells are bigger. So I think we're mostly human on a mass basis.
yes, we are. But it's more fun to talk about the cell numbers. We do also have fungal symbionts too. I think, Western people who bathe every day don't have very many, but if you lived in a more natural state, you'd probably have a lot more fungal symbionts on your skin too.
I think the public is very rapidly understanding about beneficial microbes, but maybe not as quickly about viruses.
Yeah.
Right? Because as you said,
they're scary.
the hot zone mentality is such that people are scared of them. And I think we really need to do a good job of telling people that maybe most of the viruses on the globe help us in some way.
Right.
And that's something we need to learn. I know the individuals who study phages in the ocean know that they play big roles in carbon cycles. But we still have to learn about the ones in plants and people that don't make you sick and what they're doing.
So it's not just even probably most of the people in this room still think that viruses are mostly bad. I think it's a mindset that's really pervasive among scientists too. So it's a mission of mine now to really convince people to think differently.
Who thinks viruses are mostly bad? Wait a minute.
Okay, we have a few.
Yeah, 2 people, not bad.
A few people.
This is the wrong audience.
Who before yesterday thought viruses were mostly bad? Okay, so I've done my job.
Yeah?
But I mean, in animal virology also, there have been starting to be publications about. So did you cover in TWiV the finding from Skip Virgin's lab about herpes virus conferring resistance to bacterial
we covered
infection?
that.
And, that's something, a herpes virus is something, for better or for worse, most of us have.
Sure.
And who knew that it was helping us against all kinds of pathogens?
Another related story that we covered, which many of you probably know about, is the contamination of the rotavirus vaccines with porcine circoviruses. So we talk about this, and you get feedback on the website where these episodes are posted. And you can see that people are absolutely outraged that there would be contaminants in viral vaccines, even though we don't even know if it's a problem or not. These vaccines were very well tested and there were no adverse effects. So I think that's why I asked about the plant and the viruses, because people don't want extraneous viruses in anything. Yeah. And it may be, this is a converted audience, but we have to work a lot harder to explain it.
Yes, we do.
But if they were not, I think people are afraid of genetically modified things. If it were a natural virus, a lot of the viruses you talked about yesterday or whenever it was were natural viruses. So I think, in agriculture, I'm from an agricultural region, and so I know a lot about agriculture actually, and a lot of farmers are using, bacterial infections and stuff to help modify, that infect the wasps and stuff to control insects. So there's a lot of precedent in the farming community for using microbes.
Biocontrol kind of things.
Yeah.
Well, in fact, I think probably a lot of people are using viruses and they don't know it. So we got in touch with a breeder who was trying to breed drought resistance into clover, in fact. And so we got some of their the parental breeding lines from them, and all of the drought-resistant lines had a virus in them. It was an asymptomatic virus. And their sensitive ones didn't have the virus, and they did not want to hear about this because they've been doing this breeding program for a number of years. But in fact, I'm not sure whether they're actually transferring any genes around. They think they may just be moving the virus around.
So in a situation like that, can you use the virus to infect the plants
Sure.
and make them drought resistant?
Yeah, and I suspect that's not so uncommon. Probably drought-resistant plants have been selected in a lot of places that are actually just virus infected.
Now, plant viruses are transmitted mechanically. Is that right?
Many different ways, but most commonly by insects.
Okay.
Yeah.
So if you want to artificially transmit a virus say for agriculture, how do you manage that?
Yeah, so that's something that would have to be worked out. But there are also, we would maybe look for viruses that could be seed transmitted.
Okay.
And that way, they would just be there. And that's the case for this particular drought-tolerant plant that's a seed-transmitted virus. Not all viruses are seed-transmitted, though, so.
So how do you
have to
think
pick the right
viruses? How do you think viruses protect plants?
I
from
don't
drought?
know.
I mean, we all know. I'll bet every single, every virus that people work on, induces some kind of heat shock response or another, some kind of stress response that induces, chaperones and other proteins that help to fold proteins. Maybe it also protects them from dehydration or increased osmolarity or.
Well, one thing we know about plant viruses is a lot of them induce higher sugar content in the plants. And in fact, this attracts insects to them, the insects that vector the viruses. But this may also be an osmoprotectant, too. So that's kind of the first thing we looked at. And indeed, there are a lot of sugars are elevated in these plants that are drought tolerant. But there may be multiple mechanisms, too. I hate to think that there's one unifying theme that's going to cover everything, too. So stress response is another thing. But in fact, stress response is often what kills something, not just plants, but animals too. A lot of times the pathology is due to an overreaction of your stress response system. And so viruses sometimes lower that stress response, and that's actually beneficial.
It seems to me that animal virologists need to take a cue and start trying to understand how viruses that don't kill cells or that persist might be able to help as well.
I can't believe animal virologists could learn something from plant virologists. Could it be?
That's actually a good point, and I have it here in my notes. Why do we have this segregation between plant virologists, bacteriophage virologists, and animal virologists? Why does that exist, and what could we do to
I'd like
get
to
rid
see
of
it go away. Yeah.
I mean, meetings like this certainly help, but then we go home and we go back to our viruses.
There's a lot of pressure to focus, and
There is.
And that has a lot to do with it.
But so we have a small workshop I host fairly often in virus evolution, and it's all viruses. And it's always very interesting to see how people interact that have really never talked to each other before, because this one works on phage and this one works on, herpes, and why would they ever talk to each other?
The virus structure and assembly meeting I went to a little while ago had phage, plant viruses, animal viruses. That was very integrated. That was good.
Yeah, I think it's something we should all strive for, more integration.
So one of the things we can do with our website is we put up polls. We ask people. One of the earliest polls we put up is, do you think viruses are alive? So I think one that we're going to put up out of this episode is, do you think all viruses are bad? Okay. So we'll put that up at TWiV.tv, and you can check it out. It'll have a running tally of the interaction. So do you think viruses are alive? I had the possibilities yes, no, and I don't know. And it's a third evenly divided among about 1,000 people who responded. It's very interesting. So you can do amazing things with this. This is new media where you can control it yourself. So I want to talk a little bit with Karla now. And I like one of the things you said, which we could, I think, use to start the discussion. You said, if you want to ruin a party, you have to go. And I think our listeners would love to understand what that means.
Well, when you talk at 8:30, you have to say things like that in order to wake people up. But, so what we're trying to study, we're very interested in viral evolution. And one of the applications of viral evolution is how you evolve drug resistance. And as everybody knows who works with viruses or bacteria for that matter. I mean, one of the problems with the antimicrobial compounds we have is the selection for drug resistance. So how do you fight that? It's especially poignant in RNA viruses because the error rate is very high. So that means a mistake gets made by the polymerase, 1 in every 10,000 nucleotides. So for RNA viruses, the RNA viruses a lot of us work on, poliovirus, rhinovirus, even, hepatitis C, that means, at least one mutation per genome. So drug resistance occurs fast. And, so we all collectively as a community are trying hard to learn how to control that. What's come up for HIV infection is, sure, you can have one drug for HIV, and that was protective for a while. We all probably remember when the first anti-HIV drugs came out, and people would, they'd see a drop in titer, but then in a month or two, they'd start to have resistant viruses, and so it didn't have a long-term effect. But as there get to be 2 and then 3 drugs for a virus to be resistant to all 3 of them, that's 10 to the minus 4 times 10 to the minus 4 times 10 to the minus 4. The probability is now 10 to the But you guys can do that. The probability is now very low that you have a virus that is resistant to all 3. So that's the so-called multi-drug therapy, and it's been very effective with HIV. Is that what we're going to have to do for every virus to prevent drug resistance, or are there other paradigms? And so one of the things I mentioned was other ideas are multi-drug therapy. Maybe you can choose your drug target so that the viruses that are resistant will be not as healthy. They'll be so-called less fit. A lot of us here are interested in host proteins because the theory goes, if you're infected by a virus and the virus, even if the virus is mutating really fast, if it's dependent on one of your host proteins and you don't need that host protein to live and you take a drug that inactivates that host protein, the virus will have to get over its addiction to that host protein in order to survive. And maybe that will be harder, because the host protein can't mutate not to bind the drug. Maybe it'll be harder to get over that addiction to the host protein than it will be to develop a way not to bind a drug that was directed at the virus. So that's the third way. What we're trying to think of is a new way to use sort of what we know about the genetics of viruses to say, maybe we can get the viruses to fight each other. And this is the idea. So if we're in a room right now with, 200 people in it, and if this were a cell, and we were all viruses, then, that would be sort of a typical virus-infected cell, that is, with, hundreds of genomes in it. And with the potential to interact. So if one of us, let's say Rich,
Good.
has, is unbelievably fit and has a tremendous advantage, so that if he were by himself, he could run, like a zebra all around the campus or something. But he's stuck in here with us, and there's a potential that, by just being incredibly clumsy and tripping all over the place, we could prevent you from running or even moving at all. That would be dominant inhibition of less fit individuals of your expressing your phenotype. So that's what we're trying to do for viruses. So the goal is, so we're all in this cell, right? And we get hit by a drug. And this drug inactivates us, most of us in some ways, except for now, we'll say Marilyn is resistant to this drug, right? And so depending on what's defective about us now, we may or may not interfere with her fitness. So what I'm trying to do is just figure out all the rules for that, because I think it could make the difference. We still might need multi-drug therapy, but I'd rather take 2 drugs than 4, And so anyway, so how could I interfere with Marilyn's fitness?
Good question.
Yeah. I would have to, do something. I would have to have something to do with you. I would have to be, close to you in some way. If I were sitting over there, it would be pretty hard to interfere with you. So we'd have to be, in the same room. We'd have to be close to each other. I would have to, fall down in front of you so you couldn't leave or something. So that's what I mean. In order to ruin, Marilyn's party, I have to come. I have to be there, and I have to be associated with her. In biology, this happens a lot. Classic dominance is usually when proteins are in large multimolecular complexes. And so to make a dominant inhibitor of such a complex, it has to join the complex. It can't just float around somewhere else and not join the complex. It has to bind. And so that's what I mean when we're thinking about molecularly, how do we turn, one of my proteins by binding a drug into something that's going to interfere with a more fit protein of somebody else's? It can't just unfold and just go lie around in the corner of the cell. It has to still come to the party, and then it has to just be really horrible.
Right.
So that's what we're trying to understand.
So the targets that you would select would be ones that oligomerize or multimerize.
Yes.
More or less.
Or that affect something that multimerizes.
Okay.
And, there are proteins that are multimeric that tend to just hang around in a little region. So they're multimeric, but they don't really mix subunits. So even if, we were as close as Marilyn and I were, and if I made a protein that was oligomeric, but I just kept it really close to me so that I made all the subunits myself, they wouldn't bother yours.
Right. Yeah.
So it has to not only be something multimeric, but something that I disperse. All those things aren't really first principles. That's what makes it an experimental thing as well as a theoretical.
So of all the antivirals we have a lot for HIV and some for others. Do you know if any of those have that property? Have you looked?
Any of the HIV drugs?
Sure.
It's possible that the protease inhibitors do. That's a dimer, right? And it's a symmetrical dimer around an interface. And the drugs target that interface. And it's been interesting to look at it. I'd be interested if anybody here has studied these protease inhibitors. I've been trying to follow the literature, but the interface is symmetric. And the first inhibitors of that interface were also symmetric. The later generation ones have become asymmetric, so that, if you had a mutation on one half of the protein, it wouldn't work to give you resistance on the other half of the protein. So I think people are taking advantage of the dimerization of the HIV protease, whether that's conscious or not, I don't know.
Right.
That could be partly true. And then I think, there's exactly one almost FDA-approved inhibitor of picornaviruses, which is, they were called Win compounds because they were developed by Sterling Winthrop, and then they discontinued that and fired everybody in the discovery channel, but they're still called Win compounds. And they bind to to capsid proteins, which are highly oligomeric, right? 60 copies each of each capsid protein, and there's 60 binding sites. So if all of us are, again, viruses in this cell, and we're sensitive to the Win compound, except for let me see, I'll pick somebody else.
Vincent, you haven't picked Vincent
Yeah.
yet,
Who
right?
wants to be resistant to Win compound? Yeah, you're resistant to the Win compound, and you're making perfectly good capsid protein, and if you were left to your own devices, you'd be fine, and the cell that you infected would make lots of Win-resistant virus. But we're all here too, and we're making drug-sensitive capsids, and they're mixing with yours, and so you're stuck. You can't express your fitness advantage. So I think one of these is called pleconaril, one of these Win compounds. And it was, there was an attempt to develop it as an anti-rhinovirus drug, and it reduces symptoms from 6 to 5 days. But, that wasn't enough to be approved by the FDA for rhinovirus treatment, but it's the only effective picornaviral drug we have. And actually, there's been a really exciting development that's supported by the NIH, which is one of the problems in poliovirus eradication is going to be at some point we have to stop vaccinating. And what happens, if, your baby gets vaccinated 1 week and my baby, is after the cutoff, and it plays with yours, that your baby's still going to be excreting poliovirus. And so it's really hard. This cutoff is hard. So we need antivirals to help the eradication effort. And there's been a lot of effort from the NIH put into Win compounds that are cross-reactive with all the poliovirus serotypes. So I was just hearing here that that's going ahead.
So I think this is brilliant, this idea. And I just wonder if the people who make the screen and make compounds also think it brilliant. Do you have any sense?
Well, you start talking viral genetics to these people and their eyes kind of roll back in their heads. So, one of my goals is to just have a lot of simple examples so that it's easier to understand.
Do you have any sense for how long it would take for this to have any impact?
Well, what we're doing now, so I'm trying to arrange experiments in the lab so that they're not so theoretically challenging. And so one of the things we've done actually is we've bought a lot of the first line, the drugs that are now in trials for hepatitis C, and some that are being developed for dengue. And some of them are 2 oligomeric targets. And so we're putting them all through this drill. Let's select drug-resistant variants, let's grow them all up to titers so we can co-infect at a high enough multiplicity infection, that's number of viruses per cell, that we can ensure that there's co-infection. Right. And just ask, the wild-type virus, who bothers you? Ask the drug-resistant virus, does drug-sensitive virus bother you? And that's going to be different for different drugs. And so I think maybe once we start using the drugs that are actually out there, now that there are drugs out there for hepatitis C virus, we might be able to make a little more traction with this. And my goal is to really establish these principles, if they hold up, so that we can use them for viruses that there isn't so much money in, right? Like dengue and other viruses that affect, markets that don't have as much money.
So we're going to, I think, move on to do some emails. But before we do, if anyone has a question, you can come down here. It can be related to anything we're talking about or anything to do with podcasting. Great.
So I had a question about, earlier we were talking about the drought-resistant cloves, and you were talking about, or clover, sorry, not cloves, clover.
Cloves are good.
Oh, yeah.
They're quite good, but, and you were talking about the possibility that they're, in their breeding, rather than actually moving around the genes for this, they're moving around the virus. I was wondering what the possibility might be that they're moving around susceptibility genes that allows the virus to then infect, and that leads to the drought resistance or the traits that they're
very
thinking of.
Interesting question. I hadn't thought of that, but that's a great question. Yeah, it's quite possible that that's what's happening. And I think since I first talked about drought-resistant plants and viruses, I've had a lot of plant virologists tell me that they've seen something similar to this, but they just didn't think about it. So I think it's a fairly common phenomenon, and hopefully we'll be able to delve into it. But yeah, it's quite possible that those plants that are susceptible are going to do better.
Thank you. Okay, listen to this first email from Destiny. Destiny's from California. I'm an avid listener of TWiV and TWiP. We have a second podcast on parasites, and I'm very grateful that you take the time each week to create these podcasts. They're a great learning tool. I did have several questions I'd like to ask. My first has to do with HIV. It is my understanding that currently doctors use combinations of 3 different antivirals to treat and manage the infection, and this is due to HIV's ability to evolve quickly and become drug resistant. My question is, why does HIV evolve so quickly compared with other viruses? Or does it only seem faster because the immune system can't fight it off? What would happen if you increased the combination to 4, 5, or even 10 antivirals? I'll bet you'd like to answer that.
Well, there's a myth out there that HIV evolves faster than other viruses. I mean, I think it's a myth. I mean, people have measured the error rates. There's actually some pretty careful work that's been done recently and compiled in a review article that's coming out that I probably shouldn't mention because it's in press. But yeah, I think that's a myth, but I mean, HIV is a pretty stealthy opponent. I mean, the combination of a high error rate and the ability to develop a permanent infection is somewhat it's kind of restricted to retroviruses, right? And I think Destiny probably knows about the differences between error rates of RNA and DNA viruses, but they're orders of magnitude different. I mean, it's 10 to the minus 4 for RNA viruses, and actually it ranges from about 10 to the minus 7 to 10 to the minus 11 for DNA viruses, and that is interestingly really a function of genome size. It seems like there's an optimal error rate that is such that you have, a couple mutations every generation, so that ensures variability in addition to fidelity. So. Right. So I think error rate itself is something that selection acts on. Yeah.
And I would assume that if we went to 4 or 5, 10 antivirals, it would get better, right? I mean, well, I don't know if you want to take 4 or 5 or 10.
My understanding is that with 3 you do pretty well, and the problem is if people stop taking them for a while, and then you have resistance. But if you stay on the 3, I mean, the frequency is, as you said, -4 times -4 times -4, and at 4 or 5 begins to get into logistic problems and side effects.
And side effects, yeah.
Well, also, I think they change normally people are on 3, and then they'll change to a different regime of 3 over time. So, yeah.
So her second question is, since we know that viruses evolve and become drug resistant, is there any way to predict the way a certain virus will evolve such that we can try to prevent or prolong drug resistance? My guess is probably not, but it would be nice if we could.
Ta-da! Yeah, actually, that was my brother.
Yeah, I think you can just listen to this episode and answer that one. And her last question is actually for everyone. My last and final question has to do with a podcast I heard a while back where you mentioned that a lot of immunologists and or virologists are retiring soon. I don't remember. Do you remember that? I'm in the process of studying for the GRE, and I plan on applying this fall for immunology programs. Since you know the field better than I, which ones would you recommend? I should mention that my interests right now would be for an immunology program with emphasis on virology. If I could, and I actually would absolutely love if I could become a double doctor and combine this degree with an MD. I find the human body's ability to fend off invaders and heal completely fascinating, such that I would be more than happy to devote my life to studying it. Thanks for all that you do, Destiny. Oh, there are plenty of good immunology, virology programs, right? Anybody know any? Give Destiny shout one out. Anyone? UAB. UAB, good.
University of Alabama, Birmingham.
Emory? Wash U? Michigan.
Go ahead, you can name your own program. It's okay.
All right, those are good. Ohio State. All right.
Racaniello Lab.
There's always out there doing a postdoc, something different from what you did your doctorate in. Many people do that.
Sure, absolutely. Okay, the next letter is from Mary. And this is Mary Ewell, who you may know is a blogger along with Elio Schechter. It's a blog called Small Things Considered. And Mary writes, I particularly enjoyed this week's chat with Graham Hatfull, as I am a phage enthusiast. He certainly gave the phage their rightful due, and I appreciated the mention of the blog that Elio and I write, Small Things Considered, as well as Graham selecting Forrest Rower's book, Coral Reefs in the Microbial Seas, as his pick of the week. Also in the pick of the week, you enthusiastically praised the great breadth of information contained in John Ingraham's recent book, March of the Microbes. I have my own copy close at hand here. When I received it, I turned eagerly to Chapter 10, Viruses, my favorite topic. He, like TWiV, focuses on those that make us or other animals sick. Not a word in that chapter about the phage. Of course, he has to be selective as he is covering a vast territory in one book. However, he does not simply ignore them. On the first page of that chapter, he sets the tone by quoting 2 Nobel laureates. First, David Baltimore, who said about viruses, quote, if they weren't here, we wouldn't miss them.
Whoa. If they weren't here, we wouldn't be here either.
I picked this email particularly for this. That is followed by Peter Medawar's quip describing a virus as a piece of bad news wrapped in a protein. Both highly quotable quotes, but considering the ecological and environmental role of viruses in general and phage in particular, his view of viruses seems incomplete. Would you, Rich, Alan, and all care to share your thoughts about this? All the best, Mary.
That's what this episode's been about, basically.
Exactly.
Those are incomplete. I think, in particular with the metagenomics, now that we can look at, see viruses that don't cause pathology, the perspective is really rapidly changing, I think.
I hope so.
Well, there are articles from before. I remember there's a really good review article that was written by Luis Villarreal called with the catchy title "Viruses, Sex, and Motherhood," in which he describes how in wasps they have they're infected by a virus that helps with oviposition, so it helps with oocyte development, and that's actually been coming out in mammals too. That some of our endogenous retroviruses are really involved in the process of life itself. So,
in fact, it's a lot of people think that endogenous retroviruses are why placental mammals evolved, and there is quite a bit of literature. Frank Ryan has written some popular press books. His most recent one is called Virolution, and it's a great book, even though the cover was picked by some publisher that didn't know anything about viruses. So don't be turned off by the cover of the book. It's a really, really good book.
Okay, the next one is from Jack. And Jack is sitting right there. That's Jack Morris. I was really happy to get this. Hi, Vince. I discovered TWiV recently, and I have become a big fan. Keep it up. I've been teaching an undergraduate introductory virology course for over 30 years, initially 10 at UC Berkeley and now at Nebraska. TWiV will become a requirement for my course this fall. We have some excellent virology programs here, and we recently consolidated all human, animal, algal, and plant virology into one building. And he sends a link for that. And that's great. That's a great step into solving the problem we were discussing before. I've been going through all the TWiV episodes this summer. I'm a plant virologist, and I recall one episode in which the discussion focused on innate immunity. It turns out that plants have an innate immune system with toll-like receptors and MAP kinase cascades. There is also an adaptive immune layer, RNA silencing. This is what we work on. It might be a fun topic for a future TWiV. Why don't you come on and do it with us, okay? I'm also interested in Dick's enthusiasm, that's Dick Despommier, for vertical farming. I don't know if anyone's heard of this, but my colleague has envisioned building hydroponic farms in skyscrapers. I read all his essays, and it sounds promising. What struck me, somewhat ironically, is the potential virology connection. One of the major threats to greenhouse food production are plant viruses. Commercial operations have gone belly up because they have not been able to successfully control the spread of viruses. Most problems result because the viruses are contact transmitted through roots and soil. Interestingly, some of these viruses, like tomato bushy stunt, the first icosahedron to be structurally resolved, Steve Harrison, pass right through the human gut and become contaminants in waterways used for irrigation. All quite interesting biology. Keep up the good work. You're doing a great service to the discipline.
Well, listening to Marilyn, it occurs to me that if you're going to have a successful vertical farm, you're going to have to populate it with an appropriate, an assortment of microbes.
I would agree, yeah. And I hope that we'll be able to define that more clearly in the near future.
Our next one is from Caleb. And I received this last night, and we usually get 3 or 4 a day. And I have to read this. Hello, TWiV guys. I'm in the 6th grade, and I love your podcast.
Oh, yay.
It's amazing, and it's not the first one. I have a few questions. Okay, anybody can answer these. What is the biggest virus?
Mimi.
How big is that?
Really big.
So
You can see it in a light microscope.
I think it's 750 microns, micrometers, something like that, right?
Bigger than
and for Caleb, it infects amoeba.
Right. Next question. What are viroids?
They are small infectious RNAs that don't encode any proteins. Very found in plants quite a bit. Not found in animals, but there's a viroid-like thing that's found in association with hepatitis B virus.
My favorite viroid is called gadang.
Yeah.
That affects
Coconuts.
Coconuts, and it means dying.
Gadang.
Yeah.
It means dying, yeah.
Dying.
Great.
Yeah.
Well, if it didn't mean dying, it'd be a good name for a cat.
It kills coconuts, by the way.
His next one is, what is a prion? It's a 6th grader. I mean, that's pretty good, right?
Yeah.
I mean, of course, maybe it's not a 6th grader. Maybe it's one of you. Who wants to define prion? A rogue infectious protein, and it causes degenerative neurological diseases, right? Can be transmitted from person to person, or animal to animal, or animal to animal. We had an interesting TWiV about the one in deer, chronic wasting disease of deer. It's really good. So we'll put a few links for you about these. Oh, his last one is, what is a virophage?
Sputnik.
I don't really know what a virophage is. Does anyone know?
It's a phage inside the Mimi virus.
Right.
It's a dependovirus.
Exactly. It's a dependo.
Exactly.
A virus of a virus.
Yeah, so it's a virus that depends on having another virus infect the same cell for its replication. And they're associated with the large Mimi-like viruses, right? So those are Caleb's questions. Great questions, Caleb, and you should know that they're being answered by a very expert audience here. He's from Texas, San Antonio, Texas. So we get email really from all around the world. We just happen to use ones from the U.S. today. Should we do one more?
Sure. I think we got time.
Mary. I don't know where Mary's from. I really enjoy your podcast as a scientist that trained in virology during my PhD and postdoc years. Unfortunately, I have not really been able to use the virology skills in my career since then, and I really miss that. Listening to TWiV lets me refresh and renew my virology knowledge base for one day when I may get the chance to go back to doing virology. I'm still trying to listen to all the old podcasts, but I'm wondering if you have ever talked about JC virus. I know this virus is troublesome in those with AIDS, and I lost a family member to a JC virus brain infection due to what is still being called a, quote, rare, unquote, side effect from CD20 monoclonal therapy, Rituxan. I know that Rituxan is being used for many more indications, so it seems like a little more research and in-depth knowledge on this virus would be a good thing. I hope you talk about JC one day soon. Keep up the good work. I will keep listening.
So we touched on JC in a couple of older episodes, TWiV 26 and 41, and Vince, you wrote a blog on it, but we should do a whole episode on this.
Right. I think it's very interesting. And as these monoclonal therapies, which are immunosuppressive, are used more and more, you see reactivation of many of these viruses which are there and not doing anything, and in an immunosuppressed person they do. So I think that's great. And if anyone is an expert, send us an email.
Yep, let us know.
Well, we'd love to have guests on the show. All right, let's move on to a couple of picks of the week before we wrap it up so you can get on to your next session here. Let's start with Marilyn. What have you got for us, Marilyn?
Well, my pick of the week is a paper that came out in Nature, and it's about looking at the phage, essentially, the fecal microbiota of monozygotic twins and their mothers. So they looked at all the phage from the gut of, I think there are 4 families of mothers and identical twins, and they looked at the populations 3 times throughout the course of a year. And the bottom line is that they found that there was a lot of variation between family members, but within an individual, there wasn't a lot of change over that course of a year.
This is in the phages only,
In the
right?
phages only, yeah.
So this is a very interesting question that has been brought up, and we've heard how the gut microbiome is important, but within them, the phages that lysogenize them probably have a great deal to do with that. So I think this is the start of a lot of work in that direction. So that's great. Yeah, that paper just came out. It's really neat. Karla, you got something for us? No? Okay. Rich, what have you got?
Well, my pick was sent to me by my good friend and ex-TWiV guest, Grant McFadden, who, if you get on he's got his own little spam list, okay, that you can actually subscribe to. And he'll send you stuff that interests him. And,
Can you put me on that list?
This is high quality spam, okay? And this one is produced by the American Museum of Natural History, and it's a YouTube clip. And it's an animation that shows you everything in the known universe that's been charted. Okay? So it starts with the Himalayas on Earth and pans out from that, so you see the Earth. And you pan out from that, and you see all the satellites around Earth. Pan out from that, and the moon, the solar system, etc., out to quasars or whatever, which I think is the furthest thing out they can see, and then all the way back. And it's fascinating. It's very good. And apparently there's a bunch of people who are putting together this database. It's basically 4-dimensional. All the stuff in space and with time at the American Museum of Natural History, and they have a display there, and this is a video that I guess will probably be updated as time goes on and they complete it. And it's really fun to watch.
Cool. Can I get on that spam list too? Yeah. Great. Thanks, Rich. I have a book today that was originally recommended by a listener, Jesper from Sweden. It's called The Red Queen. Anybody ever read The Red Queen? Yeah, it's great. Sex and the Evolution of Human Nature by Matt Ridley. Matt Ridley's a very good science writer. And of course, the name refers to the character in Through the Looking Glass who has to keep running to stay in the same place. And that's evolution. It's about why sexual reproduction is the best strategy for evading parasites. So it's a really good read. He takes a historical view of the field, really well written. He's a former scientist himself, and humor along with it. Humor always helps learning. And so I recommend it highly, The Red Queen. So that about does it for TWiV. Let me tell you how to listen to TWiV if you don't already know. We're on the iTunes marketplace, which is free. Apple has a wonderful clearinghouse for music as well as podcasts. And you can go there and subscribe for free, and you get every new episode as we upload them. It's also on the Zune marketplace for those of you who have Zunes. Or you can listen with your smartphone, like an iPhone or a Droid or BlackBerry. Missing any? Palm, with an application called Stitcher Radio. And it's a free app which you can download here. I think you've got it here, right?
Yep.
And it will stream the content to your phone so you can walk around campus and listen to TWiV. Okay? And so it doesn't store them, it just streams them to your device. You can always go to the website, twiv.tv. You can play the episodes there. You can download them. We have all the show notes. We have all the emails, as well as our picks of the week. It's quite an archive now that we're at episode 92. We're also part of several other websites where you can find high-quality science. We're part of Microbe World. We're part of sciencepodcasters.org and promednetwork.com. And as we always say near the end, always send us your questions and comments to twiv@twiv.tv. And I want to thank everyone for joining us today. Karla, thanks so much.
Thank you. It was fun.
Karla's at Stanford University Medical Center. Marilyn, thank you for coming.
Thank you. This has been really fun.
The Noble Foundation. And Rich is at the University of Florida, Gainesville, which I understand is the home of the Fighting Gators.
Well said. That's right.
Good to see you.
Great to be here always.
And thanks to the American Society for Virology for making this happen. We're really grateful to be able to show you what we do every week. Special thanks to the present and former chairs of the Education and Career Development Committee, Becky Dutch and Glenn Rawl, for supporting TWiV at ASV. And many thanks to our local host, Michelle Hardy, for making this happen. Of course, thank you guys. I just wanted our listeners to hear how many people are here. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.